So as a biologist, kind of my broad research interests are basically how do we start out as a single cell getting a set of chromosomes from our mother and a set of chromosomes from our father, and how do we go from one cell to more than 1 billion cells, and every single cell getting an exact set of those chromosomes from our mom and our dad. So it all comes from our genes. And so many of my college students would actually probably say that denim genes actually are part of their identity, which for them may be true, but I'm actually thinking of the genes that are made up of DNA. Okay, so hopefully you've all seen a picture like this at some point showing the nice double helix of DNA. So all the information comes from those little ladder rungs in the middle, okay? And stretches of those are what make up genes, and it's different between every person, okay? So my actually larger question here is how does this DNA get packaged up into a cell, and how does it basically get copied and then put into those two daughter cells? So when it divides, how do you make sure that every single chromosome gets copied and put into the daughter cell? So this is actually pictures showing cells as they go through replication. They go up and those chromosomes condense, and then they line up and one set gets put into each cell, and it's incredibly precise. Okay, like I said, you go from one cell to 1 billion cells, and it almost never makes any mistakes. And part of the key is the fact that there's one little spot on your chromosome that makes sure that it gets attached and pulled to the other cell, and that's called the centromere, and that's what I study. Okay, so it actually has little tubes. They look like little straws that come in and suction to the chromosome and then pull each one apart, and you can see them getting pulled to each side there. One set to one cell, the other set to the other. So this happens over and over and over, going all through development, every time your cells divide, and make sure that every single cell gets exactly one chromosome from your mother, one chromosome from your father, and this is actually what human chromosomes look like. Everybody has 2 sets of all 23 pairs of chromosomes. Like I said, incredibly precise. Okay, all because of that little structure, the centromere. But as you imagine, it's actually tough and kind of ethically challenging to always study human chromosomes. So I actually study yeast. So these are yeast cells. A lot of you may have heard of the baker's yeast or the brewer's yeast, and that's what this is, Saccharomyces cerevisiae. And of course, it has some really cool uses. For example, making wine. It's the brewer's yeast. It's incredibly powerful at taking sugar. You can just pump in sugar, and it'll take that sugar and convert it to ethanol. It'll actually keep converting it to ethanol until the ethanol concentration gets high enough that it actually starts killing the cells, which is what's great about beer and wine. It also can take sugar, and it will convert it into gas. Okay, and we use this, and this is why it's the baker's yeast. Okay, it'll convert it into carbon dioxide, and that's what causes bread to rise, and that's why you see all those little holes in the bread. Okay, so it's a little metabolic machinery that we've made good use of. Although I actually don't study the baker's or brewer's yeast, I actually study this yeast, which is just a little bit bigger, and you'll notice there's some longer filamentous cells in there called hyphae. And those cells are actually really important because that's why it can, that's part of the reason why it can actually cause disease. So all of us probably have it growing and living in our mouths, but only if, so this is my one slide showing a baby with thrush. Okay, so Candida albicans can cause certain diseases such as thrush or other more serious diseases, which is part of the reason why I wanted to study it, because I know that chromosomes getting into the daughter cells is actually really important in the yeast cell cycle. So you'll notice it looks almost exactly the same as the human cell cycle did. So that's what makes it great. I can have millions of cells in culture. They divide really fast. They're cheap to grow. And I can study the exact same process in a yeast. They still have a centromere, and I can still study centromeres, but I can do it in a yeast, and they actually function exactly the same. So what I wanted to know is what happens if you get rid of that really important part of the chromosome, that really important centromere. So as you can imagine, hopefully you can see up there, some of those chromosomes get left behind. If you don't have a centromere, that chromosome doesn't get attached, it doesn't know where to go. So one cell might end up with extra copies of a chromosome. Other cells may end up with too few. Okay? And as we know in humans, you'll notice down here, well, sorry, that's a little fuzzy. This person actually ended up with a third copy of one of the chromosomes. And in this case, chromosome 21, having 3 copies causes Down syndrome, which can lead to mental retardation, other types of developmental defects. Having this abnormal number of chromosomes is actually really important when it comes to cancer. Okay, this is actually a breast cancer cell line. And you'll notice this looks way different than any of the other ones we've looked at. Okay, you can have anywhere from 3, 5 copies of different chromosomes, and we actually have weird chromosomal fusions down in the corner. So I hope that by understanding how chromosomes segregate, we can actually understand cancer better. And actually, it's really important in the yeast that I study too, because if they get extra copies of certain chromosomes, they actually become resistant to some antifungal drugs, and that's what this is showing. On the left strip there is one of the drugs, and you can see it actually can grow in high concentrations of the drug. So hopefully mine is a little bit more technical, but this is my little drop in the bucket in science, and I really hope that our research can actually make contributions to the greater understanding of human development and drug resistance and disease. So, thanks.